So today's class is going to be about X-ray diffraction data collection and processing. So within the class, we're going to go from having our initial diffraction images. From there, we'll calculate electron density maps, and from there, we'll go to the structure, and we're going to do this using some crystallographic software. So I'll be switching back and forth between basically presentation mode and some live software. One bit of housekeeping here. So for the syllabus, our lecture is here on February 9th. Now, typically, the order of the lectures, Professor Yah's two lectures are usually here before the two of mine, and he covers the electron X-ray diffraction and the electron density map in great detail. And just because of the schedule this year, we had to move them around. So I'm going to cover just enough of the electron density equation so you guys will understand later. And then he will keep in mind that he'll cover this in much more detail later. This is the electron density equation. So basically we're solving for electron density indicated as rho in three-dimensional space, x, y, and z. That uses a triple summation wave function. I'll get back to the wave function in a moment, but first we're going to show how electron density relates to the models. Okay, first we're going to look at the relationship between a finished structure and the atom positions and finally the electron density. So what we have here is a typical cartoon representation of a protein. In this case, it is a trimer consisting of 3 identical subunits, and it's colored by trimer. And if I move it around, you can see the alpha helical portions as well as the beta strand portions that come together to form beta sheets, and the 3 identical proteins come together to form this trimer. So that's the overall structure, and this is how we typically see a picture of a structure in a publication. So what I'm going to do next is show the chemical structures of all the side chains. We can see them here. And then I'm going to turn off the cartoon. And now we have the chemical structures of the backbone and the side chains. Let me color that by element. So now our carbons are white, our oxygens are red, and our nitrogens are blue. Okay, so there's our chemical structure of the entire trimer. And finally, I can put away the chemical structures and simply show the position of each atom by a sphere. So these are the position of every atom in the protein. And this program called PyMol knows where to draw each of these atoms in space based on the chemical structure of the protein, and each atom has a particular XYZ coordinate which was experimentally determined by X-ray crystallography by measuring the electron density to identify where those atoms would be. All right, now I'm going to switch software so I can show the electron density maps. So what we see here is the chemical structure of that same protein we were just looking at, which is a trimer, here. And I can rotate it around, and we can see the chemical structure of all the side chains. And we can also see these red atoms here. Those represent water molecules. So water molecules are part of a crystal structure as they're bound by hydrogen bonds. And of course, they have electron density, just as a protein does, and essentially any atom. So up here in the corner of the software, we see an indicator showing X, Y, and Z relative orientation. And those are our 3 axes. And then what I can do is draw a box which shows the unit cell, which is the repeating unit which builds the crystal. And in this one corner here, we can see the origin where that little box is. So that's position 0, 0, 0 of XYZ. And XYZ come out from that with the orientation of this little indicator. So, therefore, each atom has specific coordinates that define it. And if we just choose a particular atom, let's say that one is, we can see down here, that's the gamma carbon in chain B of residue 80, which is phenylalanine, and its XYZ coordinates are 18.5, 922, 3.77. And every atom in this structure has those coordinates. Now what I can also do is zoom out a little bit, and I show what are called the symmetry-related molecules. And basically that is the repeating crystal structure of this protein as it goes through the crystal. And we can see the packing, and I could, represent the entire crystal in the computer if I felt like it. And we can see how it fills up, through and beyond the unit cell. So in my example here, you'll see that the protein of interest does not fill the unit cell. So that's a good time here to have a quick aside about the difference between a unit cell and an asymmetric unit. All right, so here's a simple example of the relationship between an asymmetric unit, a unit cell, and the entire crystal. So the asymmetric unit is the minimum repeating unit that describes the protein structure that we're interested in. And this is a subset of the unit cell. And the unit cell is the minimum repeating unit that you need to reproduce the entire crystal. So the relationship between the asymmetric unit and the unit cell is based on the crystal space group. And this is a simple example where there are two asymmetric units in each unit cell. A more complex space group, such as in the example I was just showing, which the space group is P212121, that has many asymmetric units in the unit cell. All right, so that's the relationship between the atomic coordinates and the structure. So now let's talk about the electron density map. So I'm going to turn on an electron density map for this protein. And we see these blue cages here drawn, and those represent the density. And for the most part, the density matches the model that we have there, the model of the crystal of the structure of the protein. And that should be because the model was built from the electron density that was determined experimentally. So we can see the backbone of the protein here, the side chains, this tryptophan, as well as the water molecules have their own electron density, and they're represented as oxygen atoms. Typically, we don't see hydrogen atoms in crystal structures. They just don't have enough electrons to scatter, especially at the typical resolution we get. So one thing I can do with this electron density map is I can scroll the density. And what I'm doing here is I'm not changing the data. All I'm doing is changing the level of the amount of density which is shown as the blue cages where the density is. So those areas that stay as density at the lowest cutoff have the most density, and those are typically where some of the backbone atoms and the atoms that are less likely to move around. And by being able to scroll through the electron density, this is a way we can essentially show what is a 4D plot on the screen. So the four dimensions are X, Y, and Z, the positions, and then the fourth is rho, or the amount of density at any X, Y, Z position in space. Okay, so this is a good time to show and talk about different resolution maps. So what we've been looking at is a 2 angstrom resolution structure and map. So at 2 angstroms, when we build the model into the map, we have pretty good certainty about where the backbone atoms and the side chains go. But each individual atom isn't completely resolved as to where it is. So for comparison, I'm going to turn off this one and turn on another model and map. And this is the same protein, But in this case, it's a different crystal structure and a different solution. And in this case, it's a 1 angstrom map versus the 2 angstrom map we had previously. So immediately in this case, we see that, I can again change the level of the density that I'm showing. And right away we see at the 1 electron density map the clarity of each individual atom's electron density. And at that point, each atom is clearly resolved. And in this case, the model can be built with much greater confidence at 1 angstrom resolution versus the 2 angstrom on the previous map. Okay, now we're back to our crystal structure with the 2 angstrom resolution map. And I can show another way, which we can show the depth of the density at each position. And to do that is to draw a variety of maps that each one has a different contour level and color. And now you can clearly see the depth of density at each XYZ position. So as you get more density, the maps get redder and thicker, and you can see that the higher amount of density is at the center of the atom positions in each case. So what I'm trying to show here is that 4th dimension of rho, which is electron density in the XYZ plot. Okay, to go back a step and reiterate about what I mean when we're solving the electron density equation, we're again looking at our model and map that we have here. So if I were to go ahead and remove the model, which is the structure, and put the unit cell back up with the origin right there, zoom out a little bit. Basically what we're doing with the electron density is we are calculating going through the equation to get a 3-dimensional representation of where there's positive electron density and how much there is at each XYZ position using that triple summation wave function that I showed earlier that I will get to in more detail in a few minutes. So this is basically the solution of that electron density map. And of course it will extend farther out. It's just not displayed here. All right, so we're back to our electron density equation, and I'm going to go through the equation for a little bit. So after that, we will solve electron density using experimentally determined data set. Okay, so let's look at the electron density term by term. Our first term here is 1 over V. V is the volume of the unit cell, and that's in cubic angstroms. So in this example, we have a cubic space group, and that's a simpler example where the 3 sides, A equals B equals C, and the 3 angles are all 90 degrees. So, pretty simply, if A has a value of 35 angstroms, then the volume is simply A cubed. Okay, so the next section of it is a nested triple summation of H, K, and L wave functions in 3 dimensions, X, Y, and Z. So to break that down, a simple wave function equation is here, and that's in 1 dimension. So what that solves for is f of x, which is the height of a wave at position x of the wave, and then f is the amplitude, and h is the frequency in the x direction, and then alpha is the phase, which is the position of the wave. And I have some examples of simple wave functions here. So in the top one here, the amplitude's 1, the frequency is 1, and the phase is 0. So then the difference between number 1 and number 2 is that the amplitude is increased by 3. Then the difference between number 1 and number 3 is that the frequency is increased by 3, which we see. And finally, the difference between the top one and the bottom one is that the phase is moved by 1/4, and we can see the wave has shifted phase. Okay, so since this is a summation of a periodic function, it's a Fourier summation. So at each h, which is a different frequency, we get a different individual wave equation. So this would be h0, frequency of 1, h1, h2, h3. And those are determined in the diffraction pattern, which we'll see in the next slide. And then when we sum those, we start by summing 0, 0 plus 1, 0 plus 2, finally up to the highest number of h which was obtained, and that will give us our complex wave function in one direction. And then since we have three directions, they're just summed in series. Okay, so the next term here is the structure factor, F, and there's an F for each HKL, and that is the resultant of all the waves scattered in the direction of HKL by all the atoms in the unit cell. This is the equation for it here. And the big thing to remember here is that the structure factor F are calculated from the measured intensities I. And the measured intensity I will get from the experimental diffraction pattern. Okay, so here is how we get the intensities. This is our representation of our experiment. We have an X-ray tube, generates an X-ray beam that interacts with the crystal. The waves diffract and interacts with the detector. So on the detector, we'll get our HKL reflections. So the ones that are closer to the center of the detector are the low frequency terms and the low H numbers, HKL numbers, and the higher frequency, and which are the higher resolution reflections, are farther out from the center. And we can see that here. This is a actual diffraction image. Right where my cursor is the beam center. And as we go out from the center, first we get our low frequency, low resolution information, and then we get out to medium, and finally our high frequency, high resolution information. Then if we zoom in here, and this is the beam center, which is the same as this, we can see as we come out from the center, each of the reflections has an HKL value, which we'll identify. This particular one is HKL142, and as we go out from the center, they increase 242, 342. So you'll see H increasing here. And then if we zoom all the way in on one reflection, we can see that each pixel has a value of how many photons hit it during the experiment, and we're able to determine the intensity by summing up the intensity of the peak of it and then subtracting the background. Okay, and then the last term in the electron density equation is the phase, represented as alpha. So the structure factor is a periodic function that has a phase. And phase is not directly obtainable from the measured reflection intensities. And what we have to remember is that each HKL reflection has its own phase. Hence the subset here. So there are methods to determine phases. Examples of them are molecular replacement, isomorphous replacement, and multi-wavelength anomalous diffraction, or MAD. Ya will just cover these in his lecture in a few weeks. Okay, so here we talk about our Ewald sphere of reflections. So we have our sample crystal here, our incoming X-rays, the crystal diffracts, we have diffraction beams, and through the properties of wave addition subtraction, they strike the pixel array detector, and at points that they there's a wave addition, and we actually get one of the reflections hitting the detector. We can measure it. So we have a pixel array detector in the lab, and basically that measures the number of X-ray photons striking the detector at the pixel position. So throughout the course of the experiment, we rotate the crystal, and that rotates the sphere of reflections in reciprocal space. And over time, if we rotate the crystal enough and in the correct positions, we should capture all the HKL reflections up to the point where the diffraction limit of that particular crystal is. Hopefully, if we do it right. Okay, now we're going to look at an example data set that we collected using the home source, and we're going to use this software called HKL 3000. So you can see our data set listed here. There's two parts of it. The first part has 40 images, and the second part has 720 images, and that's enough images to give us information about the entire crystal. So the goal of this exercise is to determine the measured intensity at each HKL reflection that we see. So displayed here is frame 1, and there are 760 of these in the data set. There's a couple things to point out. This white horizontal bar is just a space between the two detector chips. The software knows about it, that's fine. And then this vertical white bar is the beam stop, blocking the direct beam, and the beam center is right about here. So the first thing we need to do is to identify our reflections. So we use a little algorithm called peak search. What that does is it just looks for dark spots. We can adjust that a little bit until we get enough peaks to index it. So what indexing does is it measures the distances between the different peaks and attempts to determine what space group class it's in. So I'll index it here. Okay, when we index it, it gives a list of potential space groups and what the measured values are, and also what the values need to be to actually be that space group. So we see here for primitive hexagonal, where A equals B but does not equal C, and then A and B are both 90, or alpha and beta are both 90, and gamma is 120. It also gives a percent penalty of how the difference between the measured value and what the value needs to be in order to satisfy the requirements of that space group. So typically we would choose the highest symmetry space group, and we're going from low symmetry to high symmetry here with a low penalty. So it's likely hexagonal. Okay, so now the difference in our display here, and let me pull up the zoom window. Previously we had seen the red spots. That was what the algorithm decided to choose as what peaks were, just darker areas. Now these the green circles represent the predicted spots based on what unit cell we chose, and right now they line up. They match the prediction and the actual spots match pretty well. So the next thing we would do is refine this, and that does a least squares fit based on the initial unit cell as well as the image itself. We'll refine it a few times until it kind of pops into place and the differences stop changing. And we can see here, if I turn off the predictions and turn them back on, that the predicted spots match where the spots are. Okay, so the next thing I'm going to do is include all the variables in the least squares fit and refine that a few times. So that's our initial indexing. So at this point, we have our space group dimensions, A, B, and C, and we also have our angles, alpha, beta, and gamma. And what we can see is that based on this unit cell, the reflections are predicted to be at the spots where the yellow circles are, and we can see that that's where our spots are. So that is the correct indexing for this crystal. So now, the next step we're going to do is an integration step. And what integration does is it uses the information we've determined on the first frame about the unit cell. And if the concept is if you determine the unit cell from the first frame, as you rotate the crystal, you can predict where the reflection should be on further frames. Another thing I want to point out on here in the zoom image is that each one of these reflections is now identified with an HKL number. So this one is minus 161. Don't worry about the negatives. It's all relative. That's going to be 161 in our electron density equation later. And we can see the remainder. These all have their own HKL numbers assigned to them. Now if I zoom in farther and I turn on this integration box, basically what we're looking at here is that for each one of the reflections, zoom way in, this area in the center here, which is it's circle, but it's hard to draw a circle through all these square pixels, but that middle area, that's the actual peak. So these pixel values are added up, and then the background is this part of this box out here. So that there's a formula where we would add up all the pixels here and subtract the background to determine what the intensity for that particular peak is, and that one is 513.5. Okay, so that's frame one. Now we'll do integration, and that's pretty straightforward. Basically, like I said, it's going to use the information that it determined about the unit cell from frame one and go through all the rest of the frames and do the same assignment of reflections. Okay, so we can see here as it updates the frames and it puts the predictions on, the predictions still match where our reflections are. So it's going to go through all the frames and identify each HKL on each frame. Okay, so the integration's complete. It's gone through each frame. This graph is just showing us the difference statistics for each frame. So the first one is this chi-squared. That's basically the difference between where the prediction is and where the reflection is. And that's pretty flat at about 1, so that's correct. And then this is the difference in unit cell over the frame, over the list of frames. If that changes a lot, then something's wrong. Okay, so what that integration did was it created what's called a .x file for each one of the image files. So here's an example of one. So we see the file name and then .x. Basically, the important part of these files is that for each frame, it identifies each HKL reflection. So here's the list of the ones it found on that frame, 25, 30, 10. And then it has information for the measured intensity, as well as some background information and also some geometric information as to where this spot is on the frame. But the thing to remember is that the integration measures each diffraction image, records the HKL and what the intensity for that HKL is on that frame. Okay, so now what we have is we had 760 image files. Now we have 760 .x files that have the mathematical information for the intensity for each diffraction pattern that we collected. The next thing we'll do is scale that. And what scaling does is it brings together all the .x files, and the goal is to create a single, what's called a .csa file, and that's going to combine all the reflections from all the files into a single file that has a representative list of each HKL reflection, its intensity, and then a value for an error. Okay, so now let's do the scaling. So basically here we have our pending sets, which is the 760 files. I'm going to use this autocorrection that takes care of some of the noise. And then we have to choose a space group. Now you remember previously we chose hexagonal as a space group class. Now we have to pick a individual space group. Now I know what it is because I've already solved this structure, and in our case it is P3121. Normally we would have to go through and either try a bunch of these different options and see what the statistics look like, and there is some software that will kind of run through them all and give you a list of statistics, and then you can decide which is the best one. But I know it's that one. Okay, so let's let that run. Okay, so that's done. So let's look at what the output is. So this is our output file, output.csa. So on the header here, we have the unit cell dimensions, A, B, C, and then alpha, beta, gamma for the angles, and our space group. And then we have a list of all the reflections that it measured throughout the data set, combined from all the different frames. So here we have HKL003, and then the intensity and an error, which is in that background area. And we can see that for each one of the ones from 003 all the way up to 0051, and that would be one of our high resolution reflections up at 51. And basically we have a list of all of them from the whole data set. And that's basically our SCA file, and we've accomplished what we set out to do, which was to measure the reflection of all our HKL values and get the intensity for each one, and that'll be used in the electron density equation. Okay, so next we're going to move into another software package called CCP4. That stands for Collaborative Computational Project Number 4. And that is a protein crystallography collaboration group, started back in 1979. And basically what it is, it's software package that incorporates a huge list of different programs written by different crystallographers, all packaged together, so you can do a variety of things, almost anything you need to do. So we're going to use a few programs in that to take our SCA file that we created in HKL 3000 and process it. So the first thing we'll do is run a program which is called SCA to MTZ, and basically what that does is it will import our scaled data from DTREC, is the part of HKL 3000 that made the SCA, and convert it to an MTZ. So here we'll tell it what to do. I'm going to convert from scale pack, or actually it's, scale from scale pack. That's the part of scaling in HKL 2000. It's not anomalous data, so that won't be checked. And we're going to run this program called C-truncate, and what that's going to do is it's going to convert the intensities that we measured into structure factors, F, which we'll need. The formula for that is essentially the square root of the intensity as a structure factor. There's some other modifications to that, but for today's purposes, the square root's plenty. We're also going to keep the input intensities in the MTZ file. We'll need those for molecular replacement. And we're also going to add what's called an R-free column. So basically what that's going to do is it's going to assign a random number from 0 to 19 to each reflection, so that 5% of it will receive the number 0. That's used in a calculation later on in our structure where we determine R-free. Basically, an R value shows how good your structure is compared to your density, and an R-free value is where it keeps a certain set of your reflections separate and doesn't refine those. And then you can compare those to your structure, and those should also be the same if your structure is correct. Okay, we're going to bring in our SCA file here. If you remember, P21212121.SCA, and we're going to output an MTZ. I'm going to add a few names to it. The crystal name, dataset name. I like to keep those simple because they'll be in the header. If you have more than one crystal that's going to get combined, then you just number them sequentially. And it's From the SCA file, it's determined the space group and the cell dimensions. And we'll also put in the wavelength, and our rotating copper anode is here. Now, the estimated number of residues in the asymmetric unit, we don't know that quite yet, and we'll determine that in a minute. So what I'll do is go ahead and run that, and let it run, and it's finished. So now we can look at our files from the job. The SCA file we saw earlier. And let's just take a quick look at it again. Here it is. Each HKL intensity, sigma intensity, which is the background at that point. And we're going to look at our MTZ file. Now, there's a lot of information in the MTZ file, and it kind of breaks it down so we can look at it carefully. The general tab, again, cell dimensions, space group, resolution range, number of reflections included. And then it tells us about our column labels. So far we have HKL, free R flag, and then I mean, sig I mean, which are intensity and error of intensity, and then our structure factors F and sig F, which are based on intensity. Here they are, where each column is listed, and there's some statistics for each column. Minimum and maximum, you see here for the R free, 0 to 19, how complete each one is. And then we can look at the individual HKLs. So this is similar to our SCA file, where we have columns HKL. Now we have a free R flag column. We still have our intensities, and we have our structure factors and errors. Okay, the next thing we'll do is use a program to determine the Matthews coefficient. The Matthews coefficient is going to tell us about our cell content, how many of our proteins are in the asymmetric unit. So what we'll do is we'll load up our MTZ that we just created and tell it to read the parameters. So again, we know the space group, unit cell dimensions, and the resolution. Okay, next we'll need to know the molecular weight of the protein, and we can estimate that from the sequence file. There's some other options too. I have the sequence file because since I made the protein and crystallized it, I know at least what I think is in there. So if we look at that sequence file, it's in FASTA format here, and here's our sequence, 117 residues, and that way it'll be able to determine the molecular weight. And we get an output here. Figures out our cell volume, and then gives us, based on the theoretical number of molecules in the asymmetric unit, what the Matthews coefficient would be, what the percent solvent content would be. Most crystals have protein crystals have a solvent content, which is water, around 50%, and then a probability that this number of molecules in the asymmetric unit is correct. And in this case, two different ways of calculating that probability. And in this case, we see that 3 molecules in the asymmetric unit is correct. So that'll allow us to continue now that we know that the 3 should fit in there. So now we'll use a molecular replacement program called Phaser. So basically, what this does is we give it our MTZ file. Now our MTZ file has intensities, which we'll use, but it doesn't have phases. So if you remember when we want to do the electron density calculation, we need the structure factors, which are based on the intensities, and we also need the phase for each HKL. Phaser is going to help us determine phases based on molecular replacement. So to get this going, we'll give it a name, load our MTZ in, make sure it knows which columns are our intensities. It read the space group, and then we'll decide what what we're searching for. So we need a molecular replacement search model. In this case, I'm going to use chain A of a protein that is a similar protein to the one I'm looking for that we crystallized. So this is a PDB file. A PDB file is a coordinate file that describes atoms in three-dimensional space. So here, if we open a PDB file as a text file, we can see that the important parts of it are that each atom has an entry as a line. So that's our PDB file. So this basically describes the molecule that we're going to be looking for. Okay, so that's our PDB file. We have our sequence, PDB file that we're going to be looking for. Here we're going to define the composition. So this is basically running that Matthews coefficient again, but within the phasor program. So we know, here's our sequence file again, and the Matthews coefficient decided there were 3 in there, so we're going to go with 3. And then we're going to do a search. So it's going to search for chain A, which is this here, and it's going to try to find 3 copies of that. So this will run for quite a while, so we'll let that run. So basically what molecular placement does is determine phases for us. So we have an MTZ file that has our structure factors in it, but it doesn't have phases. So we're giving it an example protein. And what phasor will do is take that protein, and here we have, this is our molecular placement search model displayed in PyMol. And what it'll do is it'll place this chain within the unit cell that we gave it in the MTZ file. And it'll do a series of rotations and translations within there until it finds a position where at that position it can estimate what phases would be required in order for the structure factors in that MTZ, when you run the electron density calculation, to take those structure factors with the theoretical phases and come up with a structure that's approximately like this. And let's look at what the output files are. So it found a solution in the output files. So we have an output PDB and an MTZ and a few other files. So let's look at the PDB first. First things we want to look at is this log likelihood gain. That is the statistic of how well the molecular placement worked. Usually the cutoff for if it's a good solution is somewhere around 500 or so. So 2700 is quite good, so that's most likely right. We also get 2 Z factors, rotation factor Z and a translation factor Z. In our case here it's 8 and 13. Again, that's quite good. The scale used to be at 1 to 7 of whether it would be correct or not, so these are clearly above 7. It also reports LLG and Z scores using different statistics, but these first 2 are telling. Okay, so then let's look at the actual atoms. So we told it to search for 3 copies of a single chain. So we found 1 at least here, chain A. Scroll down, up there's chain B and chain C. So it found 3 copies. So 3 copies, good LLG, good TFC, we're good to go. So another output it made at MTZ. So this MTZ is different than previous. Now we have our HKLs, our free R flag, our structure factors, our intensities, and now we have phases. That's these 2 columns. These 4 columns are information to draw electron density maps. It's already run the electron density equation during that. But we have phases now, so we can draw maps and continue and really look at the structure of what we got. Okay, so I'm going to open the results of the molecular placement in a program called COOT, which is Crystallographic Object-Oriented Toolkit. A very useful program. Okay, so here's our electron density map by doing the electron density calculation using the structure factors that we determined from the intensities that we actually measured on the detector, and using phases that we obtained from a molecular replacement solution. So we can, you can see it looks like a mess at first, but if you look, carefully, like here, that looks kind of like a histidine side chain. You can see going in the back here, there's a large tube of density. That's probably a backbone. I'm just going to display the model that Phaser produced. Now, Phaser doesn't change the position of any atoms. All it does is on a macro scale position the PDB we gave it. So we gave it a PDB that we think is close. The molecular replacement solution was a protein from a mouse species, and we crystallized the human version of that. So it's pretty close. The sequence identity is about 95%, and we believe the overall structure should be more or less the same. But it doesn't actually change any bond lengths or angles. Phaser just places them. It's up to us to make further changes. So let me turn on the model. So here's our model in there. And you can see, at first glance, that's not too bad. You can see here this side chain fits the density pretty well, as does this histidine up here, or that's a proline in the backbone. This is our molecular replacement solution. We may see here that you can see towards the end of the density, there's some space where the solvent is, and then over here, this is actually the next asymmetric unit in the density. So this is our working one, and that's just, it'll be a continual repeating unit. So this isn't too bad. So because Phaser didn't actually change any bond angles or lengths or anything like that, this is sort of raw now. So we're going to go into a refinement program and do a least squares fit on both the positions of the atoms and the phases in a program called RefMac. So briefly, in the interest of time, RefMac is a program which does a least squares fit on the coordinates as well as the phases. And we'll look at the output in a second. So it produced an MTZ and a PDB file, like we told it to, and also another CIF file, which is a different format of the PDB. Okay, let's look at the PDB file. Okay, so here's our PDB file. Now the first thing I'm going to look at every time I run RefMac is the R and the R free values that are calculated. The R value is essentially a statistic that shows you how well your model and your map match. The R free is also how well your model and your map match, but it's only using the portion of the data that was not refined. If you're going in the right direction, these numbers should be getting lower and lower as you go, and R and R free should remain essentially equal. So as a start, directly out of molecular replacement and a single round of RefMac of 0.33 or 33%, that's pretty good. We're on our way. Closely, this is that proline I think we were looking at before. The map's somewhat better, and the map and the model matching is somewhat better. So now what we want to do to continue solving the structure is what most people do. Will do a series of manual fitting of the model to the map, followed by another round of RefMac, and then more manual fitting, and then RefMac, and manual fitting, and RefMac, until and as we do that, the model and the map will improve, and finally we get to basically a finished point, and our statistics should be much better, and we'll be done. So let's do a little manual fitting now. Now there's another map I want to pull up, right? This helps us is that areas that are green are positive difference density. That means there should be model there and there's not, and there's areas that are red. That means that we have model there, but there really shouldn't be. There shouldn't be electron density there. So, for example, in this spot, that leucine is in the wrong place, and there's a lot of green areas where we need to add something. For manual fitting, there's a bunch of tools we can use. First, I always like to have the sequence view of the protein up, so we can navigate our way around. We also need our model fit refine tools here. And we need our other modeling tools here. Okay, so now we can work. So I'll go through a couple examples of places I know that are problems. Okay, this leucine here, we can see how the side chain of the leucine is in difference density, red, and there is some electron density here and some green difference density. So if I increase the sigma level, the density, you can see that that side chain is in the wrong position. So there's a couple things we can do. We can use this real space refine function. What that does is it selects a small portion of the protein and fits it to the map. So it tried to fit it to the map a little bit better. That's not right. So we can actually manually move it around and get it into density. Okay, so that's probably better, where that green difference density is where that carbon atom is there. When we do that refinement, it'll give us some statistics when we're done if the bond angles, the bond lengths and angles and things are correct. If these are red, that means these numbers are wrong, and those are like things that chemically just shouldn't happen. You don't get, within a protein, atoms and bonds are going to be a certain way. So that's right, we'll accept that. So that's great. Okay, here's another example. Where this side chain, you can see how this aspartic acid side chain is in the very wrong place. So again, we'll use our real space refine. Kind of move that into the density where we think it is. So that's actually a pretty good fit. So visually, the model and the map are matching, but if we look at the statistics of the bond angles, they're not good. So there's a way around that. So I'm going to save that. I'm going to accept it. Oops. Then we'll use another feature called regularized zone. What this does is it enforces the bond geometry better, so without really considering the map. So that's a better bond angle fit for that whole region. If you notice, I selected the backbone, so it has more options to move. Also move this one pretty well. Okay, but we're not fitting the map well, so we're going to go back to real space refine, and that popped it back there, but our bond angles are better. So I'm going to accept that. So the combination of me manually fitting it this, and then when we go back into RefMac and we do the automated refinement, that'll fix a lot of these problems. There's one other thing I wanted to show in the maps, particular option I like, where it will create maps with multiple contouring, and this adds another dimension of electron density. So not only where it is, but you can see the depth of the density in certain places. So if we look here, you can see where we get this deeper red color within the backbone of the protein. That's where the most electron density is. And this allows you to move atoms around into the deepest part of the density. Okay, so when we think we're done, we're going to go through some of what's called validation. What validation does is it looks at different things like geometry analysis, peptide analysis, density fit analysis, differences between the non-crystallographic symmetry parts. And this is like finishing up to make sure our structure is good. One good one is density fit analysis. Here's our output from that. So we can just go along, find ones that aren't so good, and go check those out and repair those. Also, we want to look at the geometry analysis. And this is basically bond angles and lengths. And again, go through and see what's what. There's got to be something wrong with that one. Yeah, that's bent badly, so we can fix that. So we want to do all this validation for when we bring it to the PDB database. We'll have to validate it there as well. So after we've finished our structure, at some point we will be depositing it to the PDB, the Protein Data Bank. In fact, this is required for most journals upon publication of a structure. Basically, to deposit it, you'll prepare your data, basically turn the PDB and MTZ into file formats that they like, go through the validation, which you've already pre-done in Coot, and finally deposit it. And then it'll give you your finished structure, your structure ID code, and you'll be done. And finally, when we go to publish our structure, we typically use PyMol, which is a molecular graphics program, and this will allow us to draw the nice pictures that will go into our scientific journals. PyMol is a pretty deep program. You can be very creative in it. I'll just show just a little bit of basics right now due to time. So if we show our protein as typical cartoons, where we see our alpha helices and beta strands, here it is. I can color it by the 3 different chains that it found. So there's our alpha, beta, gamma subunits, and we know this is a trimer as a biological unit. We can also show it as a surface, what it looks like 3 dimensions. We can pick you can show the whole thing in cartoon, and then pick the active site and show that as sticks. Zoom in, make our figures however we like. You're only limited by your own creativity for this. So that's that. That's how we came from the beginning of the lecture. We had diffraction data from the X-ray generator, and we're all the way up to PyMol, making our pretty figures for our journal publication.